Welcome viewers. Welcome to the lecture about prokaryotic translation. Prokaryotic translation means the product that we are getting after transcription is getting converted into a protein by a method called translation. So prokaryotic translation basically comprises of three important steps. Number one is called prokaryotic translation initiation. Number two is called prokaryotic translation elongation, and the third one is called prokaryotic translation termination. To start with prokaryotic translation initiation, we will summarize the steps which are needed to initiate the process. So the important component is ribosome, which is 70S ribosome. It is a part of 30S and 50S ribosome. So these two ribosomal subunits have to be assembled together to participate in the event of translation. Second important component is the mRNA or the messenger RNA, which actually contains the message in terms of ribonucleotides that is to be read out in terms of proteins or in terms of amino acids or in terms of polymer of amino acids. And the third important component is the adapter molecule or the transfer RNA, which is involved in the translation event. We know that the ribosome has got three different sites, namely P site, A site and E site, wherein A site stands for aminoacyl-tRNA. It means here the aminoacylated tRNA or the charged tRNA enters onto the ribosome. The second important site is called as the peptidyl tRNA, wherein the tRNA binds only after peptide bond formation. And the third site is called the E site or the exit site, wherefrom the tRNA, after getting discharged or a deacylated tRNA, exits off the ribosome through this exit site. Now, there is one exception. Generally, all the charged tRNAs enter the ribosome through the A site. Only exception is the initiator tRNA, which is formilated in case of prokaryotic translation. This formilated methionine-tRNA enters through the P site. Now, the question arises that how system will make it sure that the formilated methionine-tRNA gets entry only through the P site and not through the A site or the E site, which are also equally accessible. To overcome this problem, we have got involvement of various factors. These factors are called initiation factors, denoted as IF1, IF2 and IF3. Out of this, IF1 is involved in binding to 30S ribosomal subunit onto a region that becomes the A site. It means it blocks the A site of the 30S ribosomal subunit. Similarly, we have got another factor called IF3 or initiation factor 3, which binds to the E site of the ribosome, and then we have got a crucial initiation factor called initiation factor 2, which is a GTPase. It, along with GTP, has got binding affinity towards three different sites. Number one, it binds to initiation factor 1, which is bound onto the A site. It interacts with the 30S ribosomal subunit, and above and all, it interacts with charged aminoacyl-tRNA that is getting entry onto the P site. Thereby, it facilitates the binding of formilated methionine-tRNA onto the P site. So, the formilated methionine-tRNA is lodged onto the P site perfectly, taking help of these three factors. Once these things are being done, means all the three initiation factors have done their jobs and they have made the position of formyl methionine-tRNA fixed onto the P site. Then this smaller ribosomal subunit can bind to the mRNA, as well as it now has to interact with the larger ribosomal subunit.
Ma'am, how the prokaryotic translation machinery recognizes the start codon?
Yeah, this is a very crucial question that the ribosomes have taken help of the initiation factors, and these initiation factors have make the P sites suitable for the entry of formyl methionine-tRNA. Now the question arises that how the ribosome is going to recognize the initiator codon, that is called the start codon. Here in most of the cases in prokaryotes, the start codon is AUG, that codes for methionine, but within the one transcript we can have many AUGs. Now the question arises that which AUG will code for the start codon. For that we have got certain specific interactions between the 16S rRNA ribosomal subunit, which is part of 30S ribosomal subunit. It interacts with the mRNA having a specific sequence called Shine-Dalgarno sequences. This Shine-Dalgarno sequences are mostly purine rich, and what they do, this Shine-Dalgarno sequences are interacting with the 16S ribosomal rRNA, and this interaction actually facilitates the scanning of the ribosome to get hold of the first start codon. So the first AUG that is just in the downstream region of the Shine-Dalgarno sequences is supposed to be the start codon, and the interaction between the formyl methionine-tRNA and the mRNA fixes up the location or localization of the start codon onto the P site. As I said, that formyl methionine-tRNA has to first enter through the P site, so the start codon should be there in the P site. So these interactions are being facilitated taking help of Shine-Dalgarno sequences, number one, and number two, the Watson-Crick base pairing or the hydrogen bond formation between the formyl methionine-tRNA and the start codon fixes up the position of the start codon onto the P site. Once this interaction is done, now the question comes, this bond formation between the formyl methionine-tRNA and the start codon leads a conformational change in the ribosome, 30S ribosome subunit, as a result of which the initiation factor 3 leaves the 30S ribosomal subunit, and it is very much crucial as IF3 has to be released before the 50S ribosomal subunit comes and it attaches to the 30S ribosomal subunit, because IF3 is supposed to be inhibitor of this interaction between 30S and 50S. So as soon as IF3 releases the 30S ribosomal subunit, the 50S ribosomal subunit will come and it will bind to the 30S ribosomal subunit. Once this interaction is happening, then what happens that, as I said, that EF-Tu GTP has got GTPase activity. The larger ribosomal subunit has got a site called factor binding site, wherein the GTP hydrolysis takes place. So as soon as GTP hydrolysis takes place, this EF-Tu GTP gets converted into EF-Tu GDP. And once this conversion takes place, then what happens that EF-Tu GDP has lost its affinity towards the ribosome, and it releases the ribosome along with the initiation factor 1. So the net outcome of the GTP hydrolysis is that the initiation factor 2 GDP and initiation factor 1 are leaving the ribosome. So the net outcome of this interaction is that we have got a completed 70S ribosomal subunit, wherein the ribosome is attached perfectly to the start codon, and the start codon is having the formyl methionine-tRNA lodged onto the P site. So this completes the initiation of translation. Now the system is waiting for a second charged aminoacyl-tRNA to enter through the A site, which is an usual process. As I said, that this entry is not very difficult. Elongation factors are actually involved in lodging the proper aminoacyl-tRNAs onto the A site and to carry on the process of translation maintaining the reading frame in a correct manner. So it is responsible for maintaining the rate of translation and to maintain the accuracy of translation. And in this process, it needs energy which is provided by the binding of GTP and finally involvement of GTP hydrolysis. So let's start up with the involvement of elongation factor 2, which is again a GTPase molecule. It is involved in interaction with the 3' end of all charged aminoacyl-tRNAs. What it does, it masks the 3' end of the charged aminoacyl-tRNA, thereby it is hiding off the region of the amino acid which is involved in peptide bond formation. And by masking this, it is involved in lodging the charged aminoacyl-tRNA onto the A site. Once it is lodged onto the A site, if the tRNA is lodged in a proper direction and if there exists a perfect codon-anticodon interaction, then it leads to the hydrolysis of the GTP in the factor binding region. And once GTP hydrolysis takes place, then elongation factor 2 is released. So we can say here that elongation factor 2 is actually involved in masking the amino acid which is charged onto the tRNA, so that the amino acid is not involved in interaction with any other amino acid before getting interactions or before getting involved in interactions with the mRNA. Otherwise, it may happen that the charged tRNA, without getting interactions with the mRNA, is forming peptide bonds.
Ma'am, how the fidelity of the codon recognition is ensured?
Yes, this is a very important thing to be taken care of during the translation elongation process. To minimize the errors in the translation machinery has opted for 3 different mechanisms to maintain the fidelity of codon recognition. Number 1 is we will see that there is existence of 2 adenine residues in the 16S rRNA component of the A site of the ribosome. These adenine residues are capable of forming hydrogen bonds with the minor grooves which are generated because of perfect hydrogen bonding between the codons of the mRNA and the anticodons of the tRNA. If and only if the first 2 nucleotides of the codon of mRNA forms perfect hydrogen bonds with the anticodon of the tRNA, then only these 2 adjacent adenine residues are capable of forming hydrogen bonds with the minor groove that is generated. Herein, the adenines are unable to distinguish between the AU bond or GC bond. It just focuses on perfect hydrogen bond formation. The second method to go for the fidelity of the translation process is called kinetic selectivity. Herein, we will see the GTPase activity of elongation factor 2 is exhibited only if there is perfect codon-anticodon interaction during the hydrogen bond formation between the codons of the mRNA and the anticodons of the tRNA. If there is slighter problem in this hydrogen bond formation between the mRNA and the charged tRNA onto the A site, then GTP hydrolysis won't take place and elongation factor 2 won't be released. This is the second way to maintain the fidelity. The third method to maintain the fidelity is taking help of the accommodation process. What do we understand by accommodation? Once the aminoacyl-tRNA gets entered onto the A site, taking help of elongation factor 2 GTP, it's located in such a orientation which has to be rotated in almost 70 Armstrongs to carry on the peptidyl transferase interaction, so that peptide bond formation between the amino acid bound onto the A site and the amino acids present on the P site can take place. This peptidyl transferase activity is made possible only by a method called accommodation. during this accommodation process, what happens that the tRNA onto the A site will tilt around, and this tilting generates a stress, and this stress can only be sustained if the bonding between the codons of the mRNA and anticodons of the tRNA is perfectly made. If there is some sort of mismatch there, then this mismatched codon-anticodon interaction is unable to sustain this stress and it will be released. So this is how the system has been made to maintain the introduction of perfect amino acids or to maintain the codons in a perfect manner.
Ma'am, which enzyme catalyzes the peptide bond formation?
Herein, for the first time, we have seen that the catalytic activity or the enzymatic activity is not the whole and sole property of a protein. Here, for the first time, we have seen the involvement of rRNA in providing certain catalytic activity. So, the peptide bond formation between the amino acids on the A site and the amino acids onto the P site is carried out by an enzyme called 23S rRNA component of the larger ribosomal subunit. Herein, what we see that people were arguing that maybe certain proteins are present there which are carrying on this peptide bond formation. So, structural studies were done and it was found that the active site which is responsible for the peptide bond formation doesn't contain any amino acid within the 18 Armstrong distance. Later on, it was found that in case of E. coli translation machinery, we have seen the existence of a protein called L27, but mutation studies were done with this protein by mutating N-terminal domain of L27, and it was found that mutating these amino acids have decreased the translation rate at a enormously, but the translation couldn't be, or the peptide bond formation couldn't be stopped at all. So, it was inferred finally that this protein is involved in providing a proper conformation for carrying on the peptide bond formation, but the actual catalytic activity was being provided by the 23S rRNA ribosomal subunit, which is the larger subunit of the prokaryotic ribosome. Next question comes that once the peptide bond formation takes place between the amino acid onto the A site and the peptide which is present onto the P site, what happens that the peptide bond or the peptide gets transferred from to P site to the tRNA which is present onto the A site. This process leads to a dramatic change in the ribosome, wherein what we can see that the larger ribosomal subunit has moved apart. It means the P site doesn't contains any amino acids. On the other hand, the A site is containing the polypeptide chain. So, the translocation of the translation actually initiates with the larger ribosomal subunit. Means translocation of the larger ribosomal subunit has already taken place, whereas the smaller ribosomal subunit, it means is still attached to the codons of the A site. So, to facilitate perfect translocation, what we need? We need the movement of the P site tRNA from P site to E site. Next, we need the movement of the A site tRNA from A site to P site, and finally the movement of the ribosome one step ahead to get hold of a new codon onto the A site. So, this is the basic principle of translocation. In case of translocation, what we see the involvement of another factor called elongation factor G, which also has got GTPase activity. This translocation means tilting of the tRNA onto the A site because of peptide bond formation generates a space for the binding of this elongation factor G, GTP. So, it first binds onto the peptidyl transferase center of the A site, and once it binds there, in the factor binding site the GTP hydrolysis takes place, and once GTP hydrolysis is occurring, then elongation factor G which was bound to GTP gets converted to elongation factor G, GDP, and this then gets access onto the smaller ribosomal subunit of the A site. And once it gets access onto the smaller ribosomal subunit A site, it helps in the removal of or in the movement of the tRNA which was bound onto the A site with the peptides onto them to the P site, and the vacant tRNA which was present onto the P site now reaches the E site. So this is the entire translocation process which is being carried out taking help of elongation factor G, GTP. So we have discussed about translation elongation. This process continues until and unless the ribosome gets access onto the stop codons. Once it reaches onto the stop codons, means the stop codons are lying onto the A site, then the question of translation termination comes, which is executed taking help of certain release factors denoted by RF.
Ma'am, what are the functions of different release factor?
We do have generally two different classes of release factors in prokaryotes. Number one is called class 1 release factors. They can be of two types, RF1 and RF2. And next is class 2 release factor, which is denoted as RF3. Generally, RF1 or release factor 1 is involved in recognition of stop codon UAG, and release factor 2 is involved in recognition of stop codon UGA, whereas the third stop codon is being recognized by both release factor 1 and release factor 2. So here we can see that the release factors are mimicking the charged tRNAs. As in case of tRNAs, we do have two different interaction domains. One is interaction of the anticodons with the codons of the mRNA, and second is the amino acid which is attached onto the tRNA. Similarly, release factors do have two conserved domains. In one place, they have got a conserved amino acid called GGQ, means glycine, glycine and glutamine. These residues are conserved thoroughly. Then there is another domain which is involved in interactions with the stop codons. So here we can see that we do have some peptide anticodon like elements within the release factor 1. This release factor 1, once it gets access onto the A site, what happens that it leads to the movement, or it leads to the removal of the peptide or chopping off the peptide which is bound onto the tRNA. That is, the hydrolysis of the peptide bond has to take place. Once the peptide bond is being hydrolyzed, the next question that comes is the removal of the tRNAs or the removal of the release factor 1. So the hydrolysis of peptide bond is being executed by release factor 1, and second comes release factor 3 in case of prokaryotes, is again a GTP binding protein, has got a GTP molecule attached to it, but unlike the other GTP bound proteins, it has got higher affinity towards GDP. So in its free state, release factor 3, class 2 release factor, that is RF3, bound to GDP, binds to the A site only when RF1 is present there. And what happens that in the factor binding site, it generates a alteration of the conformation when RF1 has carried on the hydrolysis of the peptide bond. So peptide bond hydrolysis leads to a conformational change in the ribosome, as a result of which what happens that the GTP molecule is being replaced by the GTP molecule. And this GTP molecule, once it reaches the factor binding site, it undergoes a GTP hydrolysis. And the energy liberated because of this GTP hydrolysis leads to release of the class 1 release factor, or RF1 or RF2, which is present there, is being released. So we can say here that once release factor 1 or the class 1 release factor is released, then what happens that release factor 3, which is bound there in the GDP state, no longer retains its affinity towards the ribosome, and it also releases the ribosome. So the net outcome of this release factors is the hydrolysis of the peptide bond, and the generated protein is released from the tRNA. Then what remains left is the existence of uncharged tRNAs onto the P site or in the E site. And finally, this is done by ribosome recycling factors. What they do, they come into the system and they are responsible for the release of uncharged tRNAs from the E site and from the P site. And therein we will see again the involvement of initiation factor 3 as we started the journey with. It comes into action and it is responsible for disintegration of the ribosome into smaller ribosomal subunit and the larger ribosomal subunit, and mRNA is also released.